Okay, should be all set. All right, then let's talk about what we are going to do today. What I would like to do today is to quickly finish off where we left off yesterday, talking about chloroplasts. All right, just to make sure you all understand what we discussed. And then we're going to begin talking about today the cell membrane, and we're going to talk about how things get into the cell. All right, how we can, we know that things have to get in and out of the cell, and how does that happen? Kevin? Okay, well, let's start off, let's go back to chloroplast first, and then we'll do all that stuff, okay? Chloroplast, real quick. Can somebody give me the job of the chloroplast? Real quick. Beth? You remember Alicia then?
Oh, John makes food.
Thank you. Makes food for what kind of things? Where do you find it? Where do you find it, Becky?
Plant cells, algae cells, and sea slugs.
Okay, well, remember the sea slugs is an exception, so don't keep that in mind. All right, it's really just the plant cells and the algae cells. All right, and what are the raw materials that chloroplasts need to make their food? To make the food for the cells? Sarah? Okay. Algae are not the same as plants because of the way that they live. All right, algae basically are singular-celled organisms or single-celled plants, in other words. Whereas a plant, when we talk about plants, are multi-celled. Okay, and so that's really the only difference between them. But yes, for the most part, they're very similar. All right, but let's go back to the question: the raw materials needed for making food for chloroplasts. Andrea? Water, sunlight, carbon dioxide. One more thing you should have in your notes.
I didn't have it.
Didn't have this part?
No,
We left off here? Okay, then let's pick it up here then. Let's write down what she has up to this point.
all we have is where you find it and where its job is.
Okay, then let's pick it up right here where we left off then, which is the next thing, is the raw materials. Raw materials. Thanks, Holly. Raw materials needed to make food. All right? And this again is the chloroplast. All right, and Andrea so far has come up with water, carbon dioxide, or CO2, that's the chemical formula for it, sunlight, and there's one more thing. It's part of the chloroplast. It's something you should have read about. We may or may not have mentioned it. Maybe think of the green color that plants have.
Oh.
Will? Chlorophyll. Okay, it's a chemical. Alright, chlorophyll. Alright, so those are the raw materials needed to make food. Okay, needed to make food. Alright, water, carbon dioxide, sunlight, chlorophyll. What is the food that is made? What do plants make? What kind of things come out of the chloroplast? Duncan?
Sugar.
Sugar. And what else? What else comes out of it? Sarah?
Is it protein?
Well, protein, no, not really. Just remember, this is the chloroplast. Okay, protein's going to be made somewhere else. Well, what else do we get from plants?
Oh.
Becky?
Oxygen?
Oxygen. And there'll be some leftover water. Okay, there'll be some leftover water that'll be, that comes out of this whole thing. Okay, so this is what chloroplasts do. Alright, the chloroplast takes water and carbon dioxide and using sunlight. What's sunlight going to provide for this process? What's the sunlight going to provide for this process? Alicia? Energy. Okay, this is going to be energy. What about the chlorophyll? What do you think the chlorophyll is going to do? Katie?
Give it color?
Well, it does give the cell color, okay? It does give the green color in plants. But what does it do in this food making process? Anybody have any ideas or any guesses? So a quick hand, Becky. What were you going to say?
Well, in the movie we saw, wasn't it? It made it move. Or I don't think that.
Well, the chorus class were moving, okay? But that wasn't what was making them move. And that's all right. It's still a good guess. How about if I tell you this? How about if I told you that the chlorophyll doesn't get used in this reaction at all, doesn't change or get used up in the reaction? I see a couple of things of recognition. Andrea? Now you're thinking of something else, but it's a good guess. At least you're thinking. Will? It's not doesn't make enzymes. It is an enzyme. Okay, this is an enzyme. Or enzymes with proteins in any case. And so chlorophyll doesn't actually do anything, or it doesn't become the sugar or the oxygen. The chlorophyll is nothing more than the place where the carbon dioxide and the water will come together to form sugar and oxygen. Beth?
I understand. So is the process like with the whole cell is all food?
Okay. What happens is this: we have the cell, big plant cell. Now the chloroplast is a structure or organelle in there. All right. Now there's usually many of them, not just one. And yes, that is where that cell is going to get its food. Okay? Because that's what the sugar is. Okay? The sugar acts as food. All right. That's where it's going to get its food to be burnt by mitochondria or whatever to give the cell energy to live. Now see, animal cells don't do this. Animal cells have to get their food from this cell. They have to get their food from the plants. All right, that's what makes the animal cell so much different than the plant cell. All right, and only plants and algae can make and use the chlorophyll. Alicia, you have a question?
Animal cells don't have chloroplast.
Animal cells do not have chloroplast. All right, the only exception to that is the sea slug, which can use a plant's chloroplast for a very short time. All right, but put it this way: animal cells cannot make chloroplast. That's probably a better way to think of it. All right, so they can't make chloroplast to have them to use. We get them from places, but we don't use chloroplast. All right, we don't have this chloroplast in our cells that can make our own food. All right, animals aren't designed to do that. Animals are designed to use the food that's already being made by the plants. Okay, that's what makes the big difference. Okay, is that clear now? Okay. All right. Does anybody happen to know the name of this process? There is a name for this big process, and I'm sure you've all seen it at least once. It's in your books, but you've probably heard of it before too. That's the only one that knows?
Well, is it?
All right, Beth, give me a guess.
No, it's not like protoplasmic.
Streaming? No, that's different. But at least that was a good guess. No, protoplasmic streams that movement again in the cell. Will? Not yet. We're going to do those in a minute. Kevin. It's part metabolism, but it's not. In other words, it works for metabolism, but it's not. That's not the name of it. Andrew? Again, that's cell division. You're all good terms, all good guesses. Is that your guess too, Jen? Let's see if we can figure out the word. Let me tell you this. Let me ask this. Can somebody tell me a word to describe the putting together of things? In other words, how can you describe, what's a name for that? Yes? Okay, combining is one way. What's another one? Something that begins with S. Jen?
Is that
Yes, this is all photosynthesis. I was looking for the word synthesis Okay, this is all photosynthesis. That's the name of this. Okay, photosynthesis is the name of this process. And it is a very, very important process. If this process did not occur, we would not exist. It's very simple. If the chloroplasts did not exist in the plant cells and the algae cells to produce the sugars and the oxygen that we need as animals, we couldn't exist because we depend on these organisms, all right, the plants and the algae, to create our food for us. All right then. Is that clear for everyone now? Does everyone feel comfortable with chloroplasts? Are there any questions about them? And this is good. With the two classes, I get confused at times where we left off. So, Jenna, we depend on it for our food and our oxygen. Yeah, our food and our oxygen. Any other questions about the chloroplast? All right then. Something else we discussed yesterday, and we want to review, or lately we've discussed, is the idea of the cell theory. In other words, the whole idea of how our organisms are built, and it falls under this idea called cell theory. And cell theory has basically three parts, three ideas. Can anybody list those for me? Anybody list those for me? You can look them up in your notes. It's not cheating this time. It's a review. Mike? All cells come from pre-existing cells. You want to do all three, or should I call on other people? Alright. How about Stacy? All living things are made up of cells. That's two. Whoever we heard from Jared. Okay. Not only are all living things made of cells, they are also the building blocks. Cells are the building blocks of all living things. All right, that cell theory, again, is very important. And as we were saying yesterday, some people may accept this, others may not. But at least in science class, at least for what we're going to discuss, this is the idea that we base our working with living things on. All right? Questions on cell theory? All right, the last thing we discussed yesterday, and it took us a long time to get it through, but the last thing we discussed yesterday dealt with the size of cells and the variety of sizes that we deal with cells. All right, and we said that most cells are what size? If you needed to come up with a term to describe the size of most cells, what would it be, Carrie?
Microscopic.
Microscopic, which means what, Carrie?
It's small, but you have to see it in a microscope.
Okay, that they're so small that you need some sort of magnifying device, a microscope, to be able to actually to see them. All right? But we said there are some extremes, all right? There are some very extreme ends to this. And that the very largest single cell that we could find is the group of cells called what? What group, Duncan? Eggs, and the largest of those is, of course, Duncan? The ostrich egg. All right, so that when an ostrich egg is very first fertilized, when it first becomes fertilized, which means it gets both sets of information, that becomes one cell, one living cell. All right? Now, the smallest cells is where we were working yesterday. And we said that if we use a human egg cell as our basis, and a human egg cell is pretty big. You can see a human egg cell with a regular magnifying lens, not even a microscope. All right? And then we said, well, let's compare the red blood cells to a human egg cell. We said, how many red blood cells would it take to line up end to end to stretch the length of a human egg cell? Yeah? 14. All right? So we know that the red blood cells are much smaller now than a human egg cell. Well, then if we take one red blood cell and we look at the smallest cells that we know about, all right, those cells are called, and I'm going to write it up on the board again so you can see it. Okay, Rickett, I want to make sure it's SIAS. Okay, no, this is the smallest cell. Okay, so if we compare these are the smallest cells. If we compare these to one human red blood cell, how many of these would it take end to end to go the length of one human red blood cell. Brendan? 35. Okay, so you can see that they're incredibly small. Incredibly small. Beth.
Okay. But anyway, with the cell theory,
Yes.
with Reinhard Koch,
Okay.
was it 1665 or 35?
65. 1665. Okay, it's good to check. Becky.
Yeah. So, aren't the red blood cells called the
No, red blood cells are...
you said the smallest cells are the red blood cells?
No, so we're comparing the small cells, these right here, to a human red blood cell. Okay, the human red blood cells are called human red blood cells. Okay? Is that clear for you?
Well, yesterday.
Yesterday it was all cracked on the end of the board. That's why I wanted to write the name up again and make sure you understood the comparison. Okay? Sarah?
You saw repetition.
Yes, I know. That's why I double-checked it and wrote it on the board. This is the correct spelling. Alright, that's why I wanted to double-check it, make sure you all have the correct spelling in your notes. Alright, yesterday we tried to cover this right at the end of class, and we ended up running short on time, and I wanted to make sure you did have the correct information. Alright? So, questions on cell size. Questions on the size of cells. Kevin? What I would know, that's a very good question. On a test, what I would know is basically what is the largest and what are the smallest. And the fact that most are microscopic. All right, that would basically cover all your aspects that you need to do. Okay? Beth?
Yeah, this doesn't have to do with the quiz. I know.
The other class, I announced the other class that they would have a quiz tomorrow dealing with this information. All right, now, I realize it's short notice, but I would also like to have this quiz occur for your class tomorrow. Now, the reason being, it's not a large quiz. It's not going to count for a lot of points. I'll tell you that right now. Okay, so you can all relax on that respect. It's not going to be a 50-point quiz or something like that. It'll probably be about a 20-point quiz. All right, and it's going to deal with the cell parts. All right, the parts of the cell. So we will have covered all the parts of the cell basically before then. If not, if by the end of class I don't feel you're ready for it, I'll announce it and you won't have that quiz tomorrow. Okay? Becky?
So is it going to count just like it's in a wake-up quiz or homework?
If it was a wake-up quiz, it would be the size of the homework. This is different. It's not a wake-up quiz. It would be a little more. No, we'll have regular quizzes too. Jenna?
Is it going to be on the silk right here that you have on the board?
It would be on, chloroplast would be included, yes. Okay? Not photosynthesis and all the rest perhaps, but chloroplast would be included.
But when, like the...
These guys?
Yeah.
No.
Okay. What about the stuff we read in the book on our own?
That would be all included on it. So basically what I would want to see on the quiz is that you know all the parts that we discussed. So in other words, cell membrane, cell wall, we read about those, at least know what they are. The mitochondria, the vacuole, the lysosomes, okay, the ribosomes, the endoplasmic, all the parts that we did our presentations on. That's what the quiz would be. And just to know them, all right, and know what they basically do. That's all that will be on. Jenna? No, not yet, because we haven't talked about it enough. Okay? All right, then. Let's then, if there's no other questions about that, I'm going to take this off. What I'd like to talk about today is I'd like to talk about the cell membrane very quickly, because we already had one group discuss the cell membrane with us. And then get into the idea of this process called diffusion and osmosis. All right. First off, let's start by simply mentioning cell membrane. All right, and we'll start by saying, where will you find, or what kind of cells will you find the cell membrane in? What kind of cells will you find the cell membrane in? Sarah? Okay.
It's both.
It's both. All cells. Okay. All right, it's both. Right, you got it at the end. All cells have a cell membrane. Okay, all cells have a cell membrane. How about location? Where on the cell will we find the cell membrane? Your group did this. Katie?
Outside of the cell.
Okay, it is the. Okay, it surrounds the cell. Okay. And what about its function or job, should we say? What is its function or job? I guess I should let one of you guys do something from your group. Brendan.
What the things the cell needs in and the things the cell something else.
Okay, so it's going to control what goes in and out of the cell. All right. Other things. Becky.
Yeah, question. Doesn't it, I was studying last night.
Yes.
Someone told me that we might have a quiz tomorrow. So, but does something with floors have to do with?
How the stuff fits in and out? Yeah, we're going to talk about that when we talk about osmosis and diffusion and all that in just a few minutes. Okay. But is there any other job that the cell membrane does, or anything else we should add? Kevin? But the cell membrane still surrounds the cell. That's why it's best not to say it's the outermost part of the cell. All right, by just saying it surrounds the cell, when we talk about cell wall, we would just stick that on the outside of it. Okay, but that's a good point. Okay, anything else about the cell membrane that we should know maybe about its job? All it does is control what goes in and out of the cell? That?
Well, I have a question.
Okay.
Okay. See, I was talking to someone in class last night, because I forgot that class too.
Yeah.
And I was studying cell membrane, and I couldn't figure out the cell membrane was different from cell membrane and cell wall.
Okay. We'll talk about that in just a second, because actually that's going to be an important comparison. All right? Let's first figure out there is another job that the cell membrane does. Actually, there are two more things it does. Alicia? Okay, obviously, protection. Okay? Protection. It's obviously another job of it. What else is it going to do? Come on, think about it. It's going to surround the cell. So what's that also going to do? Jenny?
Sort of like hold it all in here.
Right, so it's going to, if it's holding it all together, it's going to give it support. That's support. Thank you. All right, it gives it shape. Gives the cell shape, okay? But if it's going to give the cell shape, and what else is it, what should it be then? All right? Remember, this is, think about your cells. All right? All your cells have cell membranes around them. Beth?
Well, I remember, but when you touch our skin, it's flexible, so it comes back up.
So it has to be flexible, okay? We'll write that down. That's an important fact. Cell membrane, right, is flexible. Right, it allows it to have some give and some movement. Kevin? Okay, it gives the cell shape, and we can still add some space here, all right, and holds it together. Okay, you can add that to gives the cell shape and holds it together. Okay, that's what Jenny was saying too. All right, so the cell membrane does more than just control what goes in and out. It protects the cell, all right? It's the outermost defense. All right, it gives the cell its shape, it holds it together, and it also has to be flexible while doing all this. It has to be able to have some give and take. Alicia? It holds the entire cells together, right? If the cell membrane breaks, all the material in the cell, which we call what? Somebody's hand? All that material in the cell? Justin. That protoplasm, if the cell membrane breaks, that protoplasm can leak out. All right, and the cell is in jeopardy of dying. Okay, now let's just quickly to answer Beth's previous question, dealing with the cell wall and the cell membrane. All right? The differences, and I don't want to go through and list all the things about cell wall really right now because I really want to talk about the cell membrane some more. But the difference is this: cell walls are only found in plants and bacteria. Okay, so they're not found in all different kinds of cells. And the cell wall is not part of the living cell. Right? You can write it down if you're curious, okay? If you want to write it down, because it is a good fact to know. But it is something, it's in the book, and it's something when we review. Remember I told you, after we go through diffusion and osmosis, we will sit down and review all the parts of the cell again. When we do that review with the cell wall, this will come up again. But if you want to write it down now, feel free to do it, and I'll repeat. All right? The difference between cell wall and cell membrane is the cell wall is only in plant cells and bacteria. So that's one difference. All right? Cell wall is only in plant cells and bacteria. And the cell wall is not part of the living cell. And the cell wall is non-living. It's not part of the living cell. Does that mean the cell membrane is part of the living cell? Yes. All right. The cell membrane is part of the living cell, so it really is part of the protoplasm.
In the cell, in an animal,
Yes.
the outer most layer of the cell membrane instead of cell wall.
Right. And in plants, you have the cell membrane, and then the cell wall covers that. Okay. Sarah?
What cells is the cell wall found in?
Plant cells and bacteria cells. All right. Dan, is there a problem? Okay. Now, what I was going to say is this. We talk about this idea right here, where it says that the job of the cell membrane is to control what goes in and out of the cell. All right? Well, how does it do that? How can this membrane have any sort of ability to judge what goes in and out of it? Well, first off, if you think of the cell membrane, most of the time you're going to think of some sort of, solid material, like a plastic bag. All right, this is, when we think of cell membrane, you can almost think of it as a plastic bag, where it's going to surround the cell and it's going to, protect it. But like, how can material get through this? Well, it can get through it because of openings that are going to be found in the cell membrane. And these openings have a name. Does anybody know what the name of the openings is?
Pores.
Okay, they're pores. Okay, so we can write this down still in the cell membrane. All right? Cell membrane. It has pores. All right, you can just put holes. All right, it has pores. But these pores are different than, for example, the pores you'll see in the cell wall. The cell wall has pores also, because material has to get through the cell wall to get to the cell membrane to get into the plant cell. The difference is the cell membrane has pores, we're not done yet, cell membrane has pores which can be opened and closed. In other words, the cell membrane has some ability to control which things get to go in and out. All right, it's got these holes that can be opened or closed to move material in or out. Becky.
Last night I was reading, they said, my notes in my book, it said that it controls the bacteria that goes in and out, but that they don't let the bacteria, then how do we get diseases?
Okay. It's a good question. All right, the cell membrane, like you just said, it can judge what's going in and out. And bacteria, for example, which we find as being bad for our cells, cause us to get disease and to get sick. How can we get sick if the cell membrane is supposed to keep them out? Well, unfortunately, the cell membrane is not infallible. In other words, it's not perfect. And there are times when these bacteria and these bad things for our cells can get in. And that's where we get the problems, where we get disease. All right, so there are those problems that can arise. And actually, there are some bacteria, and there are some diseases, for example, a virus. All right, when we look at a virus doesn't even bother going through a pore in the cell membrane. A virus will make its own hole. It'll actually attach to the cell membrane and drill its own hole, and then send all its material into the cell. And some bacteria can do that also. They can actually attack and drill and burrow their way right through the cell membrane. So it's good, and it has some ability to control what goes in and out, but it's not totally perfect. Right? And that's why we still get sick. That's a good question. All right. Now, what we're going to do now is we're going to leave a little bit the idea of cell membranes, and we're going to talk about two processes or two ways materials get into a cell, get in and out of a cell. All right? And so, has everybody got this information so I can erase this? Anybody does? Okay. So, we're going to talk about two processes that are going to allow materials to get in and out of the cell. One of them, yes, Beth? Oh, sorry, go ahead. Go ahead.
Diffusion and asthma.
All right, well, thank you. All right, I didn't know I was going to ask that, but since you said it, that's fine. Okay, the two processes that we're going to talk about are going to be diffusion and osmosis. All right? And osmosis. All right. And diffusion and osmosis are natural ways of moving material. Let's start by saying that, right? Let's write that right from the start. All right? Both are naturally occurring ways of moving material. And what that means is very simple. The cell membrane does not have to do any work when it's using osmosis and diffusion. It doesn't have to do any sort of work to have this process occur. It occurs naturally. All right, it's one of these facts of nature that can happen. Let's do a little demonstration. All right. I have here a beaker of water. And what I'm going to do is I'm just going to illuminate it from the bottom so you can see it better. All right. That's why it's not here. It's just facing the wall. And over here, now remember we talked about before that in class when we did the microscope and stuff, we never used any sort of material like blood. All right, well, this is an exception in this case. This has been sterilized. I got a little bottle of blood here. All right. Of course it's blood. Come on, ketchup doesn't flow like that. But in any case, it's been sterilized and stuff, so we don't have to worry about it. But what would happen? What would happen to this jar of water if I was to add a drop of his blood to it? What would happen? What would flow? Why? What does it flow? Well, wait, I'm getting a lot of different answers. Let's see some hands. We'll come up with a couple different ideas here. Okay, I got a lot of ideas tossing around, but let's see some hands. Let's start with Sarah's idea. What do you think is going to happen? What's going to turn? What do you mean? As soon as I drop this in there, poof, it's all going to be ready?
No, it's just, it'll mix in. Oh,
Alright, so you think it's going to just kind of mix in slowly? Alright. Alright, John, what's your idea?
It'll hit and it'll sink.
Alright. Dribble, okay. Alright, John's saying that if it floods, should it go in? Should it drop to the bottom? Or is it going to float down to the bottom? It stays a drop.
Well, we could like deflate a little bit, but it's gonna
Okay, so I'm good. All right, Stacy.
We get like 2 like little bubbles and then it'll start floating down to the bottom.
Okay, so you think it's going to form little tiny, smaller bubbles and float down. All right. Katie, no? Okay, Will?
What happens when you pour milk in it? It sort of makes a sheet, or I don't know what do you call it. It sort of has waves in it, and it's bubble.
Okay, something whatever Will says.
Like when you pour milk in water.
Yeah. Okay, Will says it's going to look like milk in water. Alright. It's going to act like milk in water. Alright. Well, let's see what we find out. Alright, so if I drop it in. Well, wait, Becky, you have it. Alright, go ahead.
It was either going to dissolve or act like oil.
Alright, you think it could act like oil and just kind of float on the surface?
Yeah.
Alright, here we go. Let's see what we got here. Oh, wait, Justin, you have it. Sorry.
It'll go in the water and it'll spread out and turn the light.
Okay, so you think it's going to go in the water and then kind of spread out when we're like, alright, here we go. Alright, I'm going to be honest with you, it's not blood. Alright. It's not blood. Alright, I'll be honest with you, so does anybody want to change your answer to that? No? Alright. No. Alright. Here we go. Huh? It's too filled under water. Okay, here we go. Andrew, you have something else. What was it? It won't do what it did. It won't.
It'll just go move with nothing, basically.
Alright, you guys, alright, here we go. Keep interrupting, you know? What am I going to do? Alright, here we go. Everyone's ready. Alright. Okay, can everyone scare me? Alright, here we go. Alright, actually, maybe is the light too bright? Alright, now we can see a little bit. What's happening? Well, let's have somebody, let me do this. Let me add a couple more drops. Now, okay, somebody please describe what they see happening here. Somebody please explain what they see happening. Kevin? Okay, is it only floating to the bottom? Is it only floating to the bottom? Alright. Maybe see if you can, let's hear Becky's description.
Like the material was like whatever's in the air is spreading apart. Because of the water.
Okay, so you think it's spreading apart and doing what?
Like a hot.
Okay. What would happen here? I'll even add a little bit more so you can get a better chance to see it a little bit more. Alright. What do you think would happen here if I left that for 5-10 minutes? What? I want to see hands. Come on, people. We can't have everybody on at once. What are we going to find? Rachel? What's going to disappear? It's going to be clear again? It would be sort of mixed color. So you're saying what's going to happen to all this red that's in here? So what happens to the red? Where'd the red color go? Where'd the red color go? Andrea? The water's not dissolving anything. Not dissolving anything. Where's the red color going to go? Brendan? Well, it's not. Nothing's floating. We don't see anything floating. No, everything would evaporate. Katie?
It'll like go everywhere.
Everywhere. Okay, that's what's happening here. All right. What's happening is this. This, ladies and gentlemen, is diffusion. All right. This is what's happening. All right. Diffusion is a natural process. Did you see any cell membrane in here doing any work? All right. We didn't have any cell membrane doing any work in here. We didn't have any sort of special machine in here doing anything. Well, what is happening here is diffusion. All right. Now, how many people here have ever made Kool-Aid before? Okay, you all make Kool-Aid. Now, Kool-Aid, if you buy those little packages, right, if I remember my directions correctly, you take the little packet, not the one with sugar, you take the packet, you dump it in, and you add a cup of sugar, and it says to add 2 quarts of water, right? What happens if you only add 1 quart of water? What happens? Your hands, come on, hands. What happens if you only add 1 quart of water? Kevin.
It's not going to be as, it's going to be darker than
The color would be darker. What else? What else? Stacy? Okay, there'd be more sugar, so it'd be sweeter. Okay, does anybody know a word to describe this? There's a word that would describe what we would make, how it would be different from a regular Kool-Aid mix. Well?
the opposite.
Okay, what is the opposite of diluted?
Diluted.
No? What's the opposite of diluted? No, not osmosis. Nobody knows the opposite of diluted? No. Alright, Jenny? Not close. Thickening is a good word for it. Let me ask this. Anybody here ever make frozen orange juice? You've never? Who here has made frozen orange juice? Raise your hand. Frozen orange juice. Raise your hand. Okay. When you read the label of frozen orange juice, what is it called? How do they describe it? Nate? Concentrated. So if we made Kool-Aid with only half the water, it would be concentrated, right? You would have more flavor, more sugar than it was supposed to. All right? Take a look at our beaker of water here. All right? What Rachel and a lot of people have already said is starting to come through. All right? We no longer see, all right, is the water as dark or as concentrated a color as the dye? No. All right? So what, can anybody tell me what's happening in diffusion? Think about it. Everyone think about it for a minute. Try and figure out the definition. Use the word concentrated too. All right? Think about what's happening here. Here's what we started with. This is a concentrated solution, right? Because this is the dark color. This is what we ended up with. All right? It's a much lighter color. All right, Kevin, what'd you come up with? You read that somewhere. He's got a good memory, though. He's got a real good memory. Let's write that down. Diffusion, all right, is when material moves from an area of high concentration to low concentration. All right? That's diffusion. Katie. Okay, let's see what would happen if we add a little bit more. Okay, is it better with the lighter now? Okay, for me it's not because I get a glare. But all right, if I add some more, all right. Now, as I add that in there, I can't see that. Too much better. We'll leave for now. As I add that in there, where's the high concentration of color? In here. Okay, it's these streams of the dark color. That's the high concentration. Where are the low concentrations of color? Sarah?
Where it's not so dark.
Where it's not so dark. So you can actually see the process of diffusion happening right now, right in front of you, because this color, right, which is concentrated in the dark areas, is diffusing. It's moving from the areas where it's high to the areas where it's low. It's a natural process. That's the definition of diffusion. All right. Now, how many people had turkey dinner for Thanksgiving? All right. Now, let me ask this. How many people were awake and saw their mother put the turkey in the oven? All right. Most of you did. Those of you that did not, were not awake, and saw your mother put the turkey in the oven, how did you know you were having turkey for dinner? I mean, it doesn't matter that maybe your mother got a free ham or something. How did you know, even though you didn't see her put the turkey in the oven, how did you know you were still having turkey for dinner? Dan?
Should have read before.
Yes, maybe she changed her mind. Got up in the morning and said, we'll have Scrn for Thursday. How did you know? How could you still know? How could you still be sure that you were having turkey for dinner? John? What's that mean? What are you smelling? When you smell that turkey cooking, what are you smelling? Sarah? Well, Rachel, what are we smelling? What's in the fuse?
Turkey smell.
Not turkey smell. What's in the fuse? I heard somebody here saying. Not the turkey smell. What's in the fuse? You're smelling turkey. When you smell anything, it's a real amazing thing that a lot of times we don't realize. When you smell something, you are literally smelling or experiencing molecules, parts of that material. So when you smell the turkey, what's happening in that oven is where's the high concentration of turkey molecules or turkey material? In the oven. Where's the low concentration? Everywhere else. So what's happening is little tiny molecules of that turkey are breaking away and filling up or trying to fill up the air around you. And your nose will pick it up. That's diffusion. It's the same thing. It's the same process. So, diffusion does not only occur in liquids, diffusion can occur in solids, and it also can happen in gases too. Alright? We all produce natural gas, right? You've all had it happen at one time or another. You all know when somebody else has produced natural gas, right? But unless you're the person sitting next to the person that produced natural gas, it's not that bad, is it? Right? If, for example, Jarrett produced natural gas right now, Brendan, I'm sure, would be quite uncomfortable. However, Alicia's not going to find it that bad. Why? Why, Alicia? That's right. By the time it diffuses all the way over to where you are, it's not going to be as strong. Okay? Becky. Come on, people. Come on. Becky.
Well, when you have iced tea or something, you just put one of those little scoops in and then put a little water in it to mix it up.
Yes.
Well, how come it sinks to the bottom? I think it's supposed to happen naturally.
Okay. It's different. When you're mixing a solid with a liquid, it's a different process that has to occur too. The solid has to dissolve in the liquid first, and that's why you get that different. That's why it takes time.
So how come it's different when you put it in hot water than when you put it in cold water?
Because hot water can dissolve things faster. All right, that's the difference. See, that's why I chose a liquid and a liquid. All right, because these two things are actually equal to begin with. That's why diffusion happens nice and evenly. All right, but if I took a solid, let's say I took if I took one of those Easter egg dyes, you know, those little pills you get from Paz, and you dump that in, that would be different, all right? Because that would take longer. The diffusion would be different, all right? That's why I chose this. Jenny.
Then how come when you have stuffy nose you can't smell?
Because what's blocking you, the things that register what you're smelling, your receptors, okay, are in the back of your nose. And so if you have a stuffy nose, if you're congested, you're blocking them, so they can't, nothing can get back to there. All right, in order to smell something, here's something that you may never realize. A lot of the food that you eat does not taste as bad as you think. A lot of the food that you have problems eating doesn't taste bad, it smells bad. All right, and here's an experiment you can try. Some of you may have done this. Take a piece of onion and a piece of apple. All right, close your nose. All right, pinch up your nose so you can't smell, and then close your eyes and have somebody give you the apple and have somebody give you the piece of onion. You won't be able to tell them apart. No, you won't. Katie, I'm not talking about peeling the onion. I'm talking about eating it. All right, if you close your eyes and pinch your nose, have somebody give you the apple and have somebody give you the onion separately. You will not be able to tell them apart because if they're small pieces, so they're the same size, you won't be able to tell them apart because the real big difference when you take and you eat an apple and you eat an onion is the smell. And the smell helps you register what it tastes like.
I do that on my mom.
That's exactly it. All right, Beth.
I do that with spinach ones.
Yes.
I can still taste it.
Now, see, some foods, okay, now there's a difference, okay? Now, some foods, what you're going to experience is you have different taste receptors, basically bitter, salt, sweet, and sour. All right, and the food, the chemicals in the food have triggers that will set those off. And so sometimes you can hold your nose, it's not going to help. It's still going to taste bad. All right, but I'm saying in certain foods, and onion and apple happens to be one that works really neat. You can't really tell the difference because they don't set off any of those receptors any differently. All right, what I want to be able to show you and talk to you about now is this other process. This other process is called osmosis. All right, osmosis is the same thing as diffusion. It's real easy. Osmosis is the same thing as diffusion with one exception. Osmosis only works with one thing, and you need one other thing. Osmosis. Let's put the definition over on this side. Diffusion works with everything, but osmosis is different. I'm going to show you. Osmosis, first off, osmosis is the movement, not of all things. Right? Diffusion moves everything. All materials diffuse, basically. Almost all materials diffuse. But osmosis is the movement of only one material, and that material is water. All right? And osmosis doesn't work in the room. We don't say if we drop some water on the thing, it osmosifies, all right, it evaporates. That's different. It diffuses in the room. Osmosis is the movement of water across a cell membrane. And then the rest of the definition is the same. From high concentration, abbreviation for concentration is C-O-N-C period. High concentration to low concentration. All right, so that's the difference. That's the only difference. Now, I'd like to be able to show you a neat example of osmosis. To do that, I need a cell membrane. Anybody want to volunteer one? Okay. Well, first off, are we going to be able to see any of your cells? Okay, are we going to be able to play with any of your membranes? No. So, how about if I used one of the larger cells that's available to us, which would be what? Beth? An egg. Well, how can I get at the cell membrane? It's got a problem with it. What's around the egg? No, it's not a cell wall. There's something special around an egg. What is it?
Shell?
It's a shell. Right? So what if I could take the shell off and leave the membrane intact? Does anybody know how you can do that?
Yes.
How?
I did it last night.
What?
I was supposed to make a chocolate chip cookie.
Yeah?
And I was supposed to crack the egg, and I didn't. And just put it off.
Okay, if you peel it though, you're gonna break it. How about Brendan? Yeah, but see, I want the membrane. I want the membrane. Oh, Andrea? No, that's cheating too. Okay, the shell of an egg is made out of something called calcium carbonate, which is something that's a base, something that's affected by acids. Vinegar is an acid. So if you take and you soak an egg in vinegar for a long time, all right, this is just water now. If you take and you soak an egg in vinegar for a long time, what happens is all the shell dissolves and the membrane is left. This is a raw egg. Okay, this is not cooked egg. This is a raw egg. It bounces. Okay, it would break if you dropped it. All right, or if you poke at it, it will break. All right, you can keep this for as long as you want. You can cook it if you want. Just oh, this is not vinegar. This is water now. Vinegar, you have to keep it in about 2 days. Okay. Now you can try it at home. Yeah, you can eat this egg. Now, it may have a vinegary taste to it because sometimes vinegar will diffuse into the egg. All right? But, now listen up. I'm going to trust you guys. I'm going to be very trusting to you, and I'm going to allow this to be passed around very quickly. We only have a couple minutes. All right? Now, while they're doing that, let me show you something. Now, here's my question to you. What would happen? All right? First off, what's the main ingredient inside that egg? What's the main chemical inside that egg? No, what's the main chemical inside the egg? Now, what's the main chemical protoplasm? What's the main chemical? Jenny? Sorry. Oh, come on, something more simple than that. What's most of your cells made out of? Where? I've heard them say water. Now, let me ask you. Listen up, please. What would happen if I put that egg, or what should happen if I put that egg in pure water? Where would there be a higher concentration of water? Inside the egg or outside the egg? I want to see hands. Ladies, you gotta listen too. Come on, this is important. If I put the egg in pure water, this is what has a little bit of stuff in it. If I put an egg in pure water, where would there be a higher concentration? In the water or in the egg? Justin? Okay, anybody, who agrees with that? Alright, then obviously it has to be outside the egg, right? Because it's pure water. Inside the egg, there's all sorts of other stuff. So if I put an egg in pure water, what should happen to it? It should get bigger. So if I put an egg in pure water, okay, you can see that this egg, compared to that egg, has gotten swelled up. It's gotten bigger. Because water is being pulled, because of osmosis, into the egg. What if I put it in salt water? Where's the higher concentration of water? Inside the egg. So where's the water going to go? No. Where's the water going to go, Josh? It's going to squeeze in, and the water's going to go outside, and the egg would get smaller. Okay, this egg, if you see it compared to the other ones, it's smaller. Now, hang on a second. Big one goes in pure water. The other one goes in salt water, and you'll see the difference. Now, class is almost over. Let me just finish. All right, what we did today is we were able to... No, don't pack up. What we did today is we were able to get through and talk about chloroplasts and photosynthesis. We were able to go through and begin our discussion of diffusion and osmosis. All right, now, if you want to try this thing with the eggs, please just be careful with them. Now, please, let me get my egg back before you leave, okay? Go ahead. No quiz tomorrow.